let's talk about how PCR works. So this is what would happen in your particular COVID PCR test. There are only 3 or 4 stages of PCR. The first stage is your initiation. Okay, so that's when you have your template. In this case, it'll be your template DNA, because PCR works on DNA or 2 strands. But I'll talk about later on how you convert a single-stranded RNA virus such as COVID into 2 strands so that you can actually run a PCR. Now, in one of the very first steps of PCRs, obviously, this is a 2 or double-stranded structure here. So in the very first step, it has to denature. So what that means is that within the device, it goes up to 95 degrees. As a result of this increased temperature, the 2 strands separate, so they break apart there. The next step is what happens, what we've known as primary annealing. So we design these short primers, everybody. So for example, if you want to target a specific area of COVID, you design these short primers along the COVID genome. They attach there, complementary to their target as well. And then we add the polymerase, which is already in the reaction. The polymerase then binds or recognizes this annealing by the primer, attaches to this structure, and then synthesizes new DNA in the opposing direction. So that's the process of PCR. It happens from denaturation all the way to extension. That's known as 1 PCR cycle. And most PCRs take around about 35 to 40 cycles. So the COVID PCR test works like this, and it's a 40 cycle reaction. So just to put it in context, we make all the reactions up into a PCR tube. We mix all the components together. You know, we have the template, we have the primers, we've got the polymerase, so everything goes together into this tube. It gets all mixed up. Then what we do is we aliquot it into these wells. So the reaction itself, the PCR reaction, is really small. It's only 10 microliters. So that's 1/100th of a mil. So you can imagine that. So we aliquot out a 10 microliter reaction into this 96 well plate, and then that's placed into a PCR machine, which I'll talk about a little bit later. And depending on that, on how much material you have to start with, or how much COVID a patient has, you can get millions of copies of that particular gene, or you can have billions of copies as well. So it's an incredibly powerful technique, and it only works by the fact that you can design primers specific for the gene, and there is a cycling of the same reaction 40 times over at different temperatures. Now, so a development on that is that once you run the PCR, for example, such as COVID, you need a method in order to detect the product. Because what I showed you previously was that when you make new DNA, the machine itself can't see that new DNA. So you need to develop a way for actually visualizing that newly synthesized DNA. So what we've been doing in the lab recently is a lot of people have developed this or used this. So what happens is that they developed a probe here. So if we go down here, this is known as a probe. So the probe is specific to a target as well, or to your DNA. And it's a short oligonucleotide probe. And on the probe of the arm, the 5' arm, there's a fluorescent reporter. And within close proximity to that reporter, there's a quencher. So that reporter naturally emits energy. Because it's close proximity to the quencher, it absorbs all that particular energy. And only when we release this particular fluorescent reporter, once we cleave it off, it goes off into solution, and then it emits a fluorescent signal, which then the PCR machine can detect. So that's one method or one approach whereby we can measure newly synthesized DNA. So how does that work in the context of that PCR reaction, which I just talked to you about? So here, you've got your denaturation, so 2 strands separate. You've got your primers here as well. They bind to specific regions, and this probe also binds to specific regions. Now, when the polymerase is active, it starts to synthesize new product. The polymerase has what's known as strand displacement. So what that means is when it comes here, it hits the probe, it cleaves off the fluorescence, and then it chews away the rest of the probe in its way, releasing the fluorescence away from the quencher. So the moment it does that, it releases a fluorescence signal, which then the PCR machine can actually detect. So what you see here is a graph of fluorescence versus cycle. So again, the cycle is running through the 3 temperatures of 95, 60, and 72. So that's known as a cycle. And as you go through the cycles here, you get an increase in fluorescence, which is indicative of the fact that you've actually detected that particular product through its amplification. So I know it's a bit hard to take in, but all of this, so everything that I've discussed today, you'll be learning in the Hive lab. You'll be actually doing your own PCR. This is known as a fluorescent-based real-time PCR. So we'll be looking for the detection of the virus. The virus that we work in is HPV-16. So we'll all come together in the Hive. But if you need to, or if you have any questions, please let me know. We can address them at the end of this particular lecture. All right, everybody. Now, so we talked about PCR. We talked about, how it actually works. How are the 3 stages? You've got the denaturation, you've got the meaning of the primers, you've got an extension, which synthesizes the new DNA. Now, in regards to COVID, remember when I said it was an RNA virus and PCR only works on something that's double-stranded, all right? So if I take you through this scenario, everyone who's had a PCR test, you probably got a nasopharyngeal swab. It's put into a container. The swab hopefully picks up viral particles from your nasal cavity or cells which might have are infected with the virus. It's put in a solution with lice, the cells open and the virus open, which then releases the RNA, which is the basis for this particular PCR test. We have within that particular tube purified DNA, but remember, it's only single-stranded, and for PCR to actually work, it has to be double-stranded. Then in the process known as reverse transcription, it's another step just before PCR that allows us to synthesize new complementary DNA from the RNA. Now, the way the test works in terms of what it's picking up is it's actually picking up the envelope gene from the virus, and there's a positive control here in the PCR as well, and it's picking up a ubiquitous gene which the virus expresses, which is known as another polymerase. And then you have the test here, which is a positive as opposed to negative, and it crosses what's known as the threshold. So I know it's a lot to take in initially, because this concept is actually quite hard to understand unless you haven't done it yourself. So you'll be doing this next week, so it should become clearer. Now, I just want to talk about this particular step here known as reverse transcription. So remember, the virus that you want to detect, it is single-stranded, so you have to get it into a double-stranded configuration. So here is just a quick video. I'm going to have to share the sound with you. So I'm going to, hold on. Okay, can everybody hear me still? Yep.
Yep.
Okay, I'm going to play it now. So just bear with me. So this is the process of reverse transcription.
Reverse transcription is the process of making complementary cDNA from single-stranded RNA. This then requires RNA. A reverse transcriptase of a primer. RT experiment is also called cDNA synthesis. The steps are as follows. RNA is incubated with a primer at 70 degrees centigrade, denature RNA secondary structure. This is stored quickly enough to let the primer move to the RNA. The transcriptase DNA and buffer are added to the reaction. RT reaction is extended at 37 degrees centigrade or higher for 1 hour to allow for improvement. RT reaction is heated at 70 degrees C to inactivate the enzyme. The end result is cDNA.
So as everybody can see, this process of reverse transcription, the COVID virus is single-stranded. So through reverse transcription, what you've been able to do is actually synthesize the additional strand, which is the opposing strand, turning it into a double-stranded structure. Now, so you have this particular structure. The next process is obviously PCR, and I just really want to show you an animation just to solidify your understanding of PCR.
Reverse transcription.
The 3 main steps of PCR. Denature is the first step in the PCR reaction. The sample is heated at a high temperature between 94 and 98 degrees to separate double-stranded DNA from the strands. The next step is the temperature between 57 degrees Celsius, where the primers bind specifically to complement the sequences of both strands of the DNA. The primers are designed to work through fast targeting application. At 68 degrees 2 degrees Celsius, depending on the temperature. In this step, the enzyme is the primary molecules by incorporation of the building blocks with DMT.
Okay, everybody. So that was both reverse transcription and also how actually PCR works within those particular three stages with the denaturation, priming, and then also extension as well. Now, I just want to show you this last video. I might just put it on this screen because it's a lot easier for me to manage everybody now. So this is what we actually do in the lab, and this is how you set up a PCR reaction. So what we have here is, we've got the lady, she's working. So that's your particular kit. You have all these components there that she's telling us. You've got the enzyme, you've got the buffer, and you've got the template. And what we do now is just mix everything together like she's doing there. There's the 6-well plate there. Sorry, 96-well plate. She puts it in there and wait, she's going a bit fast. So if we go back, there's the Peltier block. She puts it inside the Peltier block. She activates the machine, and then it runs for 40 cycles. Okay, so typically that's how you set up a PCR reaction. It's quite easy to do. Once you do it a couple of times, you should be an expert at it. Now, the advantage of PCR is that you can actually detect any gene that you want from any organism. All you need to know is the sequence of that particular gene and how to actually design specific primers to detect that gene. So now what we'll do is that we'll bring it all together now. In terms of diagnostics, we talked about the COVID test and we went through PCR. Again, we've got the sample collection here. So we've got the RNA extraction, which is really crucial. We talked about reverse transcription, which allows you to synthesize the complementary DNA of the virus. Now, if some viruses are DNA viruses, so you wouldn't have to undergo reverse transcription, and then you add in the primers, you run the PCR, and then obviously you do the real-time reaction. Okay, so like I said, it allows, it's highly sensitive, and the accuracy of these PCRs are 95% and above. It is quite difficult to do because you can also understand that you could degrade the RNA of the virus. Once you degrade it and if the person's positive, then you're giving them a false negative result. So that's why it's a very difficult process. And diagnostic time is quite lengthy because it takes us, in the lab to do something like this at least 3 days. You've got to collect the virus, you've got to isolate the RNA, you've got to purify out additional RNA components. You've got to set up the PCR reaction. And the reaction itself runs for about maybe an hour and a half or so, and then you get the results, then you report it back to the particular patient.